Hey everyone, welcome to another Gold Bio video. This is part one of our two-part video where I'll be talking about alpha complementation and blue-white screening. This video is just going to focus on understanding the principles of alpha complementation, and then in part two, I'll use those principles to help you understand blue-white screening. If you're new to alpha complementation or you've just been struggling a little bit to understand how it all works, then you've come to the right place. The goal of these videos is to use some analogies that might be more familiar to us and then relate that back to the science of how this all works. So let's start out first by imagining that you just found a treasure map, but you don't have the whole map. Instead, you found one of the halves. We'll just say it's the half that's missing that iconic X. So in a sense, this is not a fully functioning map. The best it will do is tell you how to get partway to the treasure, but you're still not actually going to be able to find it. You'll need the missing second half that complements the first half of the map in order to find that treasure. Foundationally, this is part of the concept behind alpha complementation. Alpha complementation is usually carried out in the model bacterium E. coli. E. coli has this enzyme in it called beta-galactosidase that helps it break down sugars like lactose. This enzyme, beta-galactosidase, not only breaks down lactose, but it also breaks down its analogs like xgal. To do its job, though, beta-galactosidase has to be working right, where four identical monomers of this enzyme come together or tetramerize. And this might sound complicated, so let me just kind of relate that back to our treasure map. Remember, you have the torn half, right? So for it to function, you need the missing piece. Now, just imagine that 4 times over. 4 maps, each with a missing piece, and to work, we need all 4 full parts of the map to come together. I know that's kind of stretching our imaginations a little beyond a reality, but stay with me here. Going back to tetramerization with our beta-galactosidase enzyme, it just means these 4 small identical molecules are coming together to form what's called a homotetramer, or a larger molecule made up of those small identical molecules called monomers. And that's a really important term to remember, monomer. Just picture 4 identical molecules, 4 small identical molecules, all coming together to form this larger functional beta-galactosidase enzyme. Another way to look at tetramerization that could help is this reverse pizza idea. You and 3 other friends each have a really large slice of pizza, and they're all exactly the same. And to make it a complete full round pizza, you put all 4 slices together on the pizza dish in a way where it all fits together, making it a round pizza. Okay, now that tetramerization is kind of out of the way, we have to talk about the complementation part, or alpha complementation. Within each beta-galactosidase enzyme, there are sequences of amino acids. And if amino acid residues 11 through 41 are removed from the amino terminus, the monomer can't form a tetramer. They can't come together. So with our reverse pizza analogy, imagine if each of you took a bite out of that pizza before putting the slices back together. When you all do try to put it together, it really doesn't quite fit right. You're going to have holes in it, and basically it won't quite puzzle back together in the right way. I mean, yeah, the pizza slices can still go together, so it's not a perfect analogy, but it still kind of gives you an idea of what we mean with the missing parts. With the beta-galactosidase enzyme, removing the amino acid residues is like taking a bite out of the monomer. And when this happens, the 4 monomers can't come together at all. To get technical, and it's really important that we do in order to understand this all, the peptide missing these residues is called the omega peptide. So this would be like what we would call our pizza with the bites taken out of them, the omega pizza. And when that's missing and the monomers can't tetramerize, the complete functional enzyme doesn't assemble. Therefore, E. coli can't break down xgal or lactose. Researchers actually use this purposefully by having strains of E. coli used in cloning that are intentionally missing those amino acid residues. So to tie this back to something familiar, it's as if researchers were intentionally making a set of pizza slices with bites taken out of them. Then there's the other side of this whole thing, the missing piece, that piece or bite that would allow the monomer to form that homotetramer, or that is the piece that would allow our 4 small identical molecules to come together to form that larger functional beta-galactosidase enzyme. This missing piece is called the alpha peptide, and it complements the omega peptide. In other words, it's our missing piece of the map in the treasure map analogy, or it's the missing bite out of the pizza in the pizza analogy. So to recap, the intact monomer can tetramerize, forming the homotetramer, leading to a functional beta-galactosidase enzyme. But if there's a part missing in each of those monomers, they can't tetramerize, and therefore a functional beta-galactosidase enzyme can't form. Finally, if that missing part, that alpha peptide, complements with the omega peptide, or that is, we put our treasure map together, the 4 monomers can come together, forming that homotetramer, and now we get a functional beta-galactosidase enzyme. Right now, I've just been trying to build on this concept more and more, so now the next concept to sort of stack this all onto is how this all works in plasmids and E. coli cloning strains. Remember how I mentioned that researchers will use this concept intentionally? Well, in cloning strains of E. coli, within their genome when it comes to that beta-galactosidase enzyme, these strains only have the sequences for the omega peptide, or the treasure map with the missing piece. So if researchers were to transform those E. coli cells with the plasmid that contains the sequence for the missing part, the alpha peptide, then once the plasmid is in the cell, both peptides will be produced, complementation will occur, and then each of those monomers are complete with their missing pieces. And then that leads to a functioning beta-galactosidase enzyme. Going back to the treasure map, let's say you found the complementary piece, the part of the map that was missing, and that part with the X. Well, now the treasure map is functional and you can find the treasure. This is the basis behind alpha-complementation. In part 2, we're going to take these concepts and apply it to a popular technique that uses alpha-complementation called blue-white screening. So I will see you in the next video.